The next talk will be from Yerbol Kurmangaliye, where he's going to talk about circuitry and the genome in the fly brain. And here's a place where actually we do have the connectivity data. And it's really exciting to see, like when you look at what's possible in the fly, it's kind of, there's a completeness, right? That you feel like you can really start to pull out some rules that will be fun to see how they apply across species. So I'm really excited to see what you're doing. So he's an assistant professor of biology at Brandeis and a Pew Scholar. And with that, I will turn it over.
Yeah, well, thank you very much for inviting me, Ila. I'm very excited to tell what we do. So I'm new to neuroscience, to be honest. So I came from genetics and genomics, and my lab studied on this scale of this kind of axis, right, from genetic to instructions to self-organization, was definitely on a far left here. So we are studying mostly genetic mechanisms, but I hope today I'll be able to sort of a little bit free up myself and learn something else, right? Maybe we're missing something big. Well, we're definitely missing something big. So brains are complex. This is an intuitive statement. I want to start talk with playing this video. I got it from YouTube, but this is a video where we're going to fly through this stack of EM images in a small chunk of a Drosophila brain. Right, so what you see here, all these little circles, right? These are the cross sections of the neurons, right? And I hope you can appreciate how crowded, right, and dense this tissue. So, and thanks to this ongoing revolution, which is happening right now, right, and the connectomics specifically in flight, I mean, many pioneers and masterminds probably are somewhere around, like Sven is here, right, who build these tools for us, and it's a really exciting moment because now we can really appreciate how complex is a brain, right? Not just intuitively, but exactly sort of see what is complex about it. So we are lucky to have already 3 whole brain connectomes. Drosophila brain is relatively small, million times smaller than human brain. However, it turns out to be even more complex than at least I anticipated. So for now, they defined connectomics researchers that there is at least 8,000 stereotype cell types. And what is a cell type? We can discuss at the end. I think it's an interesting thing, right? Is it just operational term or is it something real and biological? Let's consider for purpose of this talk that cell types exist, okay? So in the visual system, which we studied, connectomics started slightly earlier. So one of the first kind of data sets was released 10 years ago when I just started my postdoc with Larry Zipursky and came to this field. So at that time, they reconstructed, that FlyEM project reconstructed a small chunk of the visual system. And if you remember the insect visual system, right, it's like a compound out with with a bunch of repeated units. So in this case, they reconstructed 7 columns of one neurofield called medulla. Very small chunk, 1% of that part of the brain. And this is the graph of 18 cell types from a middle column in this data. And this is a graph how they're interconnected. This is a connectivity matrix, adjacency matrix for the same graph. Rows are inputs, columns are outputs, numbers are synapses. Right? So you can see some interesting features. I mean, neural circuits are directed graphs, connections are weighted, right? You have strong connections, weak connections. The most important for my talk is that it's really sparse, right? I hope you can agree with me with that. It's very tiny space, they all intermingle. But somehow neurons, very selective. They only synapse on a subset of the available targets. Now, if we take the same 18 neurons, and then plot adjacency matrix for 18 neurons across all 7 columns, you get this graph. Along the diagonal, you can see the connectivity within the same, between the same cell types in each column. Right? I can, at least qualitatively, I hope you can appreciate that they're similar, right? So I think connectons as stereotype is actually interesting in the field. Some people look to the data and say, oh wow, it's not stereotype, and I look and I say, oh wow, it's stereotype. So probably it's somewhere in between. Let's just summarize it for now like this. The strong connections are definitely highly stereotype. At least we can find them across the brain. There's a very long tail of weaker connections, which are much more varied. And there is a large, of course, proportion of the technical noise still, but there's a lot of biological variation, that's for sure. And now we have several connectons, so we can finally kind of appreciate and decouple probably these two things. Anyway, so we call this kind of a sequence genetically hardwired, which means that probably it was somehow inherited, right? So we're born and we have this circuit. So now in parallel to connectomics, there have been this ongoing another revolution, which Anna was talking about, right? And it's really great revolution which is happening with molecular side with the invent of the high throughput single cell technologies. Now we can easily pull out essentially a molecular profile, a program for any neuron in any system. So this is a one data set from Drosophila visual system, and clusters as cell types, and they mapped with the same color to this little subgraph from the visual system, okay? So the idea here, what if we combine these two sets of information, right? What if we take the transcriptomes and map on the graph? Essentially, you'll get a data set where you have information about the genes which can be present on both sides of every synaptic image. So this is the main approach my lab is using now, and we're trying to decode the problem of synaptic target choice. So we had to solve a couple technical problems, and they just sort of probably worth mentioning. So first of all, oops, I moved too fast. Oh, okay, here. So the development has a time, right? That's a problem. So the development happens not in one moment, and Drosophila is 100 hours, and there's a lot of different steps. And of course, the genetic programs will be changing rapidly, and we needed to like atlases which cover everything. And for our system at that moment, there was only like adult, of course, maps, which makes sense. So how we solved that problem, for us, it worked really well. We took a bunch of like wild-type Drosophila strains, which we know the genotypes, crossed it to the lab strain, sampled heterozygotes at every 10% of brain development, put everything sequenced together, and there is a ULab developed, right? This is for humans. Like you can demultiplex time points just based on SNPs and the transcriptomes. In Drosophila, it works even easier because you have like 10 times more SNPs for the genomic length. And so in one shot, you can get a kind of a span of entire development. This is how the data looks like. So you have sort of a data set which you unwrap by the time point. These two clusters, red and blue, corresponds to these two cell types in the connectome. Now we can loop expression of this gene with very high temporal resolution. And I can tell you, at least for the transcriptomes, because it's much easier, right? We did a lot of these experiments. The transcriptional programs are highly stereotyped. So for example, these are like, every dot here is like one experiment, and here we have like 4 replicates, right? You cannot see really the separation. So the genetic program is pretty stereotyped. So, okay. So bigger problem actually, how do we know that this red and blue cells have these cells, right? This is actually the biggest bottleneck for the whole field, I guess, right? We have like thousands of molecular maps with thousands of clusters, most of them orphan. We don't know what are they. In Drosophila, we needed to map them to connectome. We're lucky, not lucky, right? This is the main reason why we work in Drosophila. There is a genetic reason. So we have a Flylight project from Genelia have generated released, right, thousands of genetic drivers. Each of them can target different cell types. You can buy them for like $12 each. And here are the 2 drivers. You can see they target and light up specific population in the brain, and this is the same cell types in the connectome. Right? We can use these drivers, purify neurons, and generate like a reference transcriptome, so we can then use it across data sets. To make it a little bit faster, we're using exactly the same trick. Now, instead of demultiplexing time points, we're demultiplexing genetic drivers. So we take a bunch of drivers, cross it to a bunch of panel of reporter lines, they bring in GFP, and we put just one wild-type chromosome, sequence everything together, separate it by genotypes, and now you can ask, where the neurons from this cross landed? They all landed in one cluster. This cluster was LC50, right? So super best data. So this is a kind of a real data set. Here in one experiment, we run 2 samples, 2 10x experiments, and we profile like 50 drivers. They don't have to be clean. You can have intersectional drivers. And the rows are clusters, and the columns are crosses, and we can then annotate all the cell types, right? And this is the way kind of we're using now to map the cell types. So right now we have a decent kind of a data set for the visual system. Hopefully we'll expand it to other parts of the brain. There is some additional technological kind of bottlenecks. We almost solved them. So for the visual system, for some various technical reasons, it's much easier, but the connector has 700 cell types in the visual system. It's about half of the brain by mass, but cells are repeated, right? This is repetitive circuits. And for 120, we mapped so far one-to-one to the developmental transcriptomes. So again, interesting question maybe for discussion, right? Is the one cell type in connector matched to one cell type in the molecular side? So I would say, as far as we know now, if you have enough resolution on the molecular side, it is true for 99% of cases, let's say. There is interesting cases, like it's biology, right? It's like any language, you have a rule and then you have like 20 exceptions. But in principle, it's true. And it's nice, right? That if we can find the cells in two modalities, probably cell types are real, right? It's a real biological entity. So anyways, what you can do with this kind of data sets? A lot of different things. We are specifically interested in this problem of synaptic target choice, right? So let's say you have a circuit, you know how the neurons are interconnected, you have the transcriptomes, you can look at expression of energy. So right side, I'm looking just at how the data look for a small snapshot of sets of genes. Rows are genes, call them cell types. Also we have some time. Red is high, blue is low. These are the what we call cell recognition or cell adhesion molecules. These are the cell surface proteins with some large ectodomains, with some adhesion domains, and some of these genes were already shown to be implicated in viral in terms of they give phenotypes. Yeah, and they look as we expect, right? They look like they should look from, right? Every neuron expresses different combinations with different levels, different temporal dynamics. So it might probably, if you have a molecular substrate for connectivity, it should look like that. So in principle, right, if you go and probably ask in any system, I think it's true for any system, if you ask what makes neurons different, two families of genes will be on top, right, always. Transcription factors, defining cell identities, and cell surface proteins. And this is true for mice, I believe, for primates, and for Drosophila for sure. So the problem here is that, not problem, we have the data, right? We have a problem now, kind of decoding this into the graph, right? Obviously, transcriptomes encode many different things, right? How can we now take a gene and link it to a specific connection, right? This is non-trivial because I think, and we can talk about it later, I think there is much more variation than matters actually in the transcriptomes. There's a lot of pleiotropy, only a subset of these genes matter. So how can we pull out that data? And I had this power slide for myself. Let's formulate a question, right? Zoom out and formulate a question, right? We're trying to find the logic, of course, right? Can we compress the complexity of the brain? Can we compress this genetic program somehow? And what if, right? What if there is no logic? It's possible, right? What if every connection is built with a completely idiosyncratic set of rules, which were found by random walk of evolution, right? That's also possible. And yeah, I'm going to show you some data, and you make your conclusions. So we're going to focus on one specific circuit. It's a Drosophila motion detection circuit. Pretty famous cells called T45 neurons. They reside in the middle of the visual optic lobes. And this is how they look like under the light microscope. You have this green, sparsely labeled neuron, soma here, projects to this region where it had its dendrites, received inputs, that projects back to one of the four layers of this structure we call lobular plate. I hope you see layers, that will be important. In magenta, you can see the entire population of these neurons, right? They have piled the entire visual field. It's the biggest population, actually. You have 6,000 on every side of the brain. And there is 8 flavors of these inputs. There are T4s, they receive inputs here, and T5s receive inputs in this part we call lobular. These are the on and off pathways. And then each group can be further subdivided into 4 subtypes, depending where they terminate their axons, these 4 layers, A, B, C, D. And each of these layers are tuned to the direction in one of the cardinal directions. So these are the dendrites of these neurons. If you look from the top, sort of from visual field side, the stalk is here, and they grow these asymmetric neurites anti-parallel to motion, which they compute. So we have this very interesting wiring, right? Very similar neurons, 2 types of dendrites, 4 types of axons. Now, the connectomes of this neuron is really nice. So of course I moved it manually so it looks structured, but it is structured. So here you have a T45s, and on top are inputs, outputs. This is adjacent symmetrics, okay? So for every subtype, I'm showing the 5 individual neurons grouped by subtypes. These are their partners grouped at the level of type. So for example, this 4 neurons input to all T4s, and these 4 neurons all T5s, okay? And interestingly, their axons and dendrites kind of flip orthogonally their specificity, right? So T4 and T5A then go to the same layer of lobular plate, and they synapse into downstream target with exactly the same synaptic needs, which is kind of interesting, right? You can essentially partition the synaptic specificity of dendrites and axons. So from dendrite side, it's just 2 cell types, T4s and T5s. From the axon sides, it's just 4 cell types, A, B, C, okay? So this is how single cell era started for me. So we, at the beginning of kind of this era for us, we purify this neuron sequence. And during the development one time point, not surprisingly, we got 8 clusters. If you sequence these neurons in adult animals, sorry, you got only 2 clusters, right? They converge, so they lose all the differences. So differences only exist molecular during the development. And then we find clusters, okay, now you go find markers, try to figure out what's going on. And here the surprises start. There is no marker genes I could find for any subtype. So here, the expression of 3 transcription factors, which are differential, but you can see they always express in groups of neurons. So this was strange. So I was like figuring out what's going on. So it turns out, like we do clustering, right? We do dimensional reduction first, and we do some space clustering, then reduce space. And it turns out that there is a 3 linear components, which explains almost all variability in this data set, which was interesting because, I mean, what is this linear components? It's essentially just the groups of co-regulated genes which were splitting our cells to the 2 halves. So here, for example, columns are cells, rows are genes, and this, like half of the cells were expressing these genes, and another half this. And we had this kind of a binary transcriptional modules splitting cells in halves, but halves were different. If you put it back together, you get this very nice cube, which looks like a cartoon. It's a date, right? And I never saw this. I mean, I was working in genomics all my training short career, but you know, it's like you never find something like that. It was fascinating. So, and everything started making a sense. Now, if you plot the transcription factors, now you can see what it means, right? Each transcription factor is just expressing a half of the cells. But every time half is different. And then we mapped it to the circuit. Become even more interesting. It turns out every module defines one specific wiring features of the neuron. So the first transcription factor, BI, it tells you, are you a horizontal motion detection or vertical? The second transcription factor separates these 2 big domains into 2 sublayers, which is opposing direction of motion. And the third domain, independently, tells you, are you sending dendrites here or here? So we started this study with a hope to find a molecular correlates of wiring. In this case, like the transcriptome looked like a connector. This was exciting. We didn't find more of these cases, just tell you. So, but this was the motivation that, yes, transcriptome have that information about the connector, right? So, okay, we did genetics. I forgot about this part. So, of course, it's a fly, so we need to do the genetics. So, general idea which we have, so how do you build a cell type? In this case, you can think about T45 as a sort of modular, this composite kind of organization when you will have a large pan-neuronal program. I haven't told about it, but neurons essentially, the most of the transcriptomes will be shared between neurons and separate them from other cells, right? They make them, give them ability to build synapses, all that stuff. Then you make a neuron, then you add these 5 transcription factors. They're not expressed in the exact same combination in other cell types, and they define the general class, T45s. They share most of their features and physiology properties. Then you start kind of a layering these additional layers which can specify specific domains, in this case axons and dendrites. So here you can see in green all T45s, right? You have dendrites here, and the axons and 4 layers. Now if you knock out this gene, called VI, you lose layer CD, dendrites are intact, AB is intact, CD is gone. If you knock out this gene, GRN, dendrites are fine, big 2 domains, vertical, horizontal are fine, but you don't have any more sublamination. And we showed that this is not cell death, this is a probably recoding of cell identity essentially, right? So transcription factors in principle is a very powerful tool. So it's probably easiest tool to go and manipulate circuits, and it gives you a lot of information, but it still keeps the black box too big, right? I mean, it controls large sets of genes. So the next step was, of course, to go and figure out what they control. We didn't do any kind of a gene regulatory studies yet, but now we have a transcriptome of synaptic markers, right? Now we can see what is actually happening between the pairs of the neurons. So in magenta is the cell types, which are partners of T45s and mapped to the atlas. This is our data plot. There's 10 more cell types we know identity now. So we're going to focus on these 5 purple neurons. These are the cells called LLPC neurons. So why they're cool, each of them receive an input just from one pair of T45s. And this is how they look like under the light microscope, sparse clones. So each of them have like main dendrite in one layer where they get inputs. So why they're actually cool is this. So this is the transcriptional similarity based on whole transcriptome at some one time point in development. And you can see as expected, right, all T45 cells very closely related sister cell types. They're very distinct from all other neurons. It turns out that purple neurons, 4 of them are also sister cell types. One of them is kind of a adopted sister cell type, so it's probably some idiosyncratic conversions. But this part is interesting, right? So you have 2 groups of neurons which are very similar, share probably development, and they converge in the same little part of the brain we call lobar plate, and they make all these parallel choices. They kind of differentiate and make these decisions. So if we zoom in a little bit more, we're going to focus on this half of the circuit. I'll call them like orange and green T45s. They converge on these 2 cell types, which are also most similar. These are their reconstruction of their dendrites. So they literally have dendrites and adjacent layers, and this is synapses. And we try to find kind of the simplest case, right? T45s come, and they have to make a binary wiring choices. They want to connect this one or this one. And the same region of the brain, very similar neurons. How they can do that? We go to transcriptomes, again, problem. A lot of genes are expressed, everything is kind of crazy. So these are the expression patterns. The columns are one large family of cell recognition molecules, IGSF CAMs, and these are the cell types. So it's a problem, right? Of course, every neuron expresses dozens and dozens of genes because they have to do a lot of different things. Now, how can we fish out the genes which are regulating this specific choice? So now try to look to up 4 panels and sort of just quickly scan with your eyes the patterns. You would notice that they're almost identical, actually, for most of the genes. So if you ask what is different between orange and green neurons, there is, depending on thresholds, about like 12 genes throughout the genome. And one of them is transcription factor GRN. And the top gene, this most contrasting band here, is a cell surface molecule called SIDE2. Its parallel is another top differential gene, SIDE4, is expressed in orange neurons. In both cases, these genes peak during the synaptogenesis, also was a nice sign. We can add here one more layer of information. In this case, we're adding the beat side interactome. So sites is a large family of the parallels, and they form this heterophilic binding kind of, they have a binding specificities to the members of another IGSF CAM family called beats. So originally, the family first members were found in forward genetic screen as axon guidance molecules by Corey Goodman in the 90s. And Kaizen decoded these partial interactomes. So we're lucky to have interactors for these 2 cells, bit 6, bit 2, and bit 2b. we can look where they're expressed in the circuit. And in this case, we got lucky, right? You have to be lucky sometimes, as always. But anyways, all orange neuron targets express B2A, B2B, binding partner of site 4. And similarly, LLPC3 express BIT6, high affinity binding partner of site. So we found like a perfect correlation here. We have two pairs of neurons making these parallel synaptic pathways, and they have a matching expression of receptor ligands, paralogous receptor ligands. So we did some genetics. So here you can see a wild-type T14 neuron, sparse clone. Then right here, go back to lobular plate and have a presynaptic site in magenta in layer D, as in connectomes. If we generate a homozygous mutant clones, here are the 3 clones. So this one unlabeled is a T5A. These two are T4 and T5B. So they go still to the layer B, and they make synapses as they're supposed to. But now they make additional synapses in layer C, ectopic synapses. So this is the interpretation. And then we did it from other side. You take a downstream neuron. This is a sparse clone. Dendrites are restricted to layer D, postsynaptic sites. You knock out BIT6. You get an expansion of dendritic branches. You get a postsynaptic sites like spread across both layers. So this is the interpretation of these phenotypes. And let me read it, then I'll explain. So this is a summary of not only this study, but many things emerging kind of in our field. So of course, wiring decisions cannot be specified by one gene. And there is a hierarchy of wiring decisions. T45 needs to first go to a specific neuropil, and this hierarchy of wiring decisions restrict gradually the pool of available targets. And at some point, probably all T45s, remember they express a bunch of these genes shared. They say, oh, we like LLPCs as a class. Then you add a little bit more information. They say, well, we like sort of a vertical motion detection circuit. Then you add one more bit side too, and now they suddenly see, oh, this is different neurons. I prefer this one, right? If you remove this gene, what happens essentially for D neuron? Now these two cell types are molecularly equivalent, right? So the neurons do not look like a log-key perfect match kind of relation. They're looking for the best match. Okay, so what else? Okay, this is a good part, right? One gene work, so we can like try to generalize, saying, oh, site 2 and bit 6, you express them. Now you can have a higher chance. You create biases. It will affect wiring. So this is, I can show you because it's published, so it's fine. They will not cancel our paper, no, I'm kidding. So essentially, here's the situation which kind of contradicts to that statement. So these are several bits and sites which are differential. I want to say we did the genetics 2 more, we didn't get the phenotypes. That's very typical. It's actually surprising that we get a phenotype with site 2 and we can talk why. Interesting site is here, right? So green cells express site 2, orange cells doesn't express site 2, and that's responsible for downstream treatment. Why this gene that doesn't bias any of these inputs? And I can tell you I'm not showing data, right? The 3 of these cells express BIT6. So they should have a differential adhesion on dendrite side as an axon side, but it doesn't. Somehow on dendrite side, this difference is like hidden. And we can come up with a bunch of different like mechanisms, possible mechanisms, but it's just wanted to show that it's not trivial, right? In this case, if we would try to kind of generalize the rule, we would fail, right? And it's the same neuron. Correlation works on our axons, on dendrites it doesn't. Okay. Simple, for example, explanation would be sites are mostly presynaptic and bits postsynaptic. That would be one of the explanations. Okay, now zoom out a little bit more. This is rows are now genes, columns are different cell types in the visual system. This is a glia for comparison, and these are expression of bits and sites, 22 genes. It looks like, right, it could be a molecular substrate for the connectomes, right? Every neuron expresses a unique combination. And this is just one family. There is about, depending how you want to define, if you're generous, maybe 300 genes in the Drosophila genome, which are expressed in this fashion and have this adhesion exodomains. So, yeah, we haven't solved the problem So we have this approach. We can take all these maps, and I hope I convinced you that approach works. And this is sort of a big question for my lab, right? Can we actually explain or predict the connectome from transcriptomes? So far we can, and this is sort of a more operational target, right, for our lab. Can we rewire a circuit or innate behavior in a way we want? And I think that's the direction we want to move. I mean, our field is mature and successful. Like we break circuits, great. I think the next step should be, can we actually, right, rewrite the connector? And if you think about it, right, when I was undergrad, oh, sorry, I'm out of time. Okay, this is how we understand the data. Like we know in broad strokes that the combination of transcription factors regulate combination of cell adhesion molecules. Somehow that specifies wiring specificities. Yeah, this is my lab. Collaborators, a lot of work from my postdoc lab. Thank you. Yeah.
Thanks so much, Yerbol. I think the T4, T5 transcriptome clustering is just textbook beautiful, right? And so especially the transcription factors that are clearly upstream of whatever causes these differences. And so when I go then into the quite beautiful cell adhesion molecules that differentiate the function, I note that the developmental profiles in the transcriptomes are exactly what they should be, and so this is extremely useful. But for the readouts, if I saw this right, I think you only showed adult outcomes. So the developmental processes are now becoming really interesting, right? So what are these molecules actually doing? There's a number of different ways other than, say, just selecting and matching at the time of, what might, you can call it the moment of choice or whatever. But there's so many different mechanisms how these molecules in time, and maybe that also gets us into a discussion of self-organization, can actually play out what will in the outcome then look like this perfect or stereotyped specificity. So I'm curious about to what extent you've looked at the mutant phenotypes and the ideas of how to then reprogram the wiring diagram by misexpression and combinatorial stuff and so forth. So what actually happens? Is it really a more branching phenotype and then they happen to be more available in that layer? Or is it a function of pruning that normally they're all there but then maybe later specifically certain ones go away and that's why you don't have synapses? Or is it actually a biasing at the moment of synapse formation? So all of these are on the table once we look at development, right?
Yeah, absolutely. I mean, I think that it's not a bigger problem, but the next step, right now it's all correlations and genetics. Right? We have a transcriptome, which is very upstream, and we have, as you said, final phenotypes. There's a big black box in the middle, and I think one of the problems why we cannot just look at the gene expression and predict the connectome, as you're saying, we just don't know cell biology. I think the cell biology of the wiring, where to go, right? We need to just know what this gene is made. And in terms of the developmental phenotype, I hope that will happen, at least that's being discussed. There will be connectons for developing brain, which will add a lot of information, right? Because one of the mechanisms, not simple mechanisms, but one of the ways to sort of build so many connections, right? I have the numbers there, but essentially you have 8,000 cell types, you have a 1.5 million strong connections in the connectome, and you don't have that much genes, even if you have isoforms. So you have to separate in time and space.
Yeah, exactly. So maybe it's such an important discussion that maybe I'm allowed to just very quickly follow up. Even if you have had developmental connectomes, you would not have the dynamics, right? So maybe one of the functions of these beautiful molecular interactions could be slowing down or speeding up, and that's all it takes in time to cause that phenotype. So I think there are so many beautiful possibilities what and how these molecular interactions could play out that are beautiful in the outcome, but during the time it happens, maybe just having developmental connectomes wouldn't even capture that because it's not just cell biology; it's just development happens in time.
No, I mean absolutely. I mean I'm just saying it like, of course I'm a user, I'm not doing connectomes, but it's nice to have connectomes. you know. So of course, if you can do live imaging of every neuron, that would be great, right? Yeah, I agree with you, right? There could be tons of mechanisms, and I think one of the problems, right? Less data you have, more diverse and beautiful models we can build, right? So, but we are going more and more constrained, right? We'll see. I mean, I think.
So the more data you have, the uglier the models then get. Or?
Yeah, but they will be like, we have a few of them, right? So it will be better.
No, it's perfect.
Beautiful text. First, a clarification. Those transcription factors that you showed us in the cube, those are expressed with that kind of specificity early in development? What does it mean throughout development? All the way to when they make the connection, or in the progenitors that they're dividing and making their choices?
So progenitors, I don't know. Unfortunately.
Or early postmitotic neurons, let's say, way before they have connected.
Yeah.
Is that what happens? Okay.
Once they are already postmitotic, they have these transcription factors, and actually the classical view, some people call them terminal selectors.
Yeah.
Right? And this is the transcription factors which are kind of a stay on. I don't have any data yet. So this is one time point. So that's
it's like in C. elegans, so they stay on throughout, and they're sufficient to define the final pattern of connectivity, like in C. elegans.
Yeah, it's weird that actually the transcription factors are also stable, but the programs are regulate and dynamic. So it's another interesting question. You have a specificity kind of constant, but the dynamics is set by different factors.
Right. So this connects to my question, or what is downstream of this transcription factor in the first place and during this critical event? And what would it take to model all of this, right? So one can say there are many cells, there are many genes, there is a process of development. All of these things can be profiled in a way, at least at a transcriptional level. And maybe this is a question for you, Ila. Could we get to principle by which the Drosophila wires, and do you think that those principles, and maybe this is speculative in the spirit of having this discussion, would hold as you now go to a mammalian brain, for example, where the complexity of cell type is higher, the connectivity is, I guess, perhaps more complex. We can agree or disagree on this. There is more plasticity. There are very long periods of development that don't play out here. Will the principle be the same, and can you get to principle? What is the way to get to principles?
Well, I mean, I think this is kind of the general discussion today, right? So, how much of it's like these genetic instructions versus some principles, right, of self-organization? I was trying to think.
Principle, I mean, and not principle. How does it work, right? Like you have all of these genes early on. Then what? Where do they go? And can you model to a point you can make the prediction on the connectivity, which will give you the principle by which this is built?
We'll see. I mean, I don't have an answer directly to your question, but what I mentioned slightly, right? The pleiotrope, right? I think the transcriptions heterogeneity we see is more than we should see. So there is a more genes regular. No, there is just, they don't matter. A lot of things get buffered.
Yeah.
There is interesting situation that maybe I'll show it here.
Oh, that's for sure. Like in the cortex, you see a lot of that.
Yeah, I just wanted to
Redundant, yeah.
A, B cells and CD cells. They are transcriptionally much far from each other because they come from different
pools.
organisms.
Yeah.
That the connectome distance is the same. So I think kind of a transcriptomes has some luggage from evolution and development, which is there because of pleiotropy. And actually the genes which matter will be much smaller subset. In ideal situation, you want to take a transcriptome and somehow subtract evo-devo origin actually. And I hope when it's lived will correlate more. Does it make sense?
It does, and I'm not sure I agree. But we can talk about it later.
That's the point. That's the point.
I don't know. I mean, I have no. Yeah.
In line.
Yes.
That was a great talk. Thank you. So I have a question maybe a little bit specific about.
Can you speak louder?
Yeah, I can. Is this better? So I'm interested in whether you think that the reason that you haven't yet figured out this mapping between the single cell transcriptomics and the wiring is because, can you hear me? Okay, is because the sequencing data is insufficient. Somehow things like splicing or alternative isoform usage don't show up, or because the wiring code is somehow hasn't popped up yet.
Yeah, of course. I mean, we don't have a complete data. I mean, this is what we're discussing with Robert, right? Like, essentially, we don't have a complete process, right? We don't know how things happen. We don't, like, genes are changing, right? Just in, like, more simplistic way, the connectome we see is a final product. We don't know when each of the edge of the graph appear. The very simplest approximation, that's why I was, like, saying the connectome will help, right? At least now we will have a more precise way to say this gene matter for this connection because we can separate them in time. And actually, at the very beginning when I got it, I thought I would correlate and solve it right away. Spatial constraints, right? Neurons do not see each other, actually. Like, you can be pushed out by, a neuron can be pushed out by neuron B, and that's why you cannot connect to neuron C. So there is a lot of missing information, that's for sure. Well, at least, yeah, I hope so.
Yerbol, can I ask a quantitative question? Because it seems like in Drosophila, with the, the transcriptomic data that you have and then the depth of the connectomic information, presumably you can put like specific numbers on this. You can tell us what fraction of all connectivity is explained by the transcriptome. And you can tell us that about whether, the adult connectivity and how much is specified by the adult transcriptome, adult connectivity by the, earlier stage transcriptome, and so on, right? So is it possible to put those numbers on the data? I assume one could, right?
Maybe you could. I couldn't do it. But I can tell you just numbers, right? Like, so there is 15,000 genes. So we have half of them detected in the neurons. To be honest, there will be like 1,000 only genes, probably, which are somewhat variable between cell types.
Mm-hmm.
And then we can put numbers on the, I mean, it's not numbers you're asking, but just sort of there will be like a feature space of 1,000 genes, with isoforms, maybe 5,000 genes. And then, connectome, I don't know like how to describe the specificity at this point, to be honest. Of course, we draw it as a graph, but as I said, for example, the problem of these graphs, they don't have a spatial structure, which is very important. Which neurons see each other, right? So we can get that, in principle, at least for adult. But during development, that differs. And I think just sort of, maybe it's possible, maybe I just don't have the skills. I mean, you should try it. Like, how much variability is explained, right, essentially? I think the variability of the connectome, how to quantify connectome is another bottleneck. Because the specificity, actually, there is an interesting paradox. Defining specificity turns out to be very hard because it's a mutual relationship, which is unfair. You can be a partner, and for you, your synaptic partner is like top partner, 50% of your connection. For that neuron, you doesn't matter. You're like 5% of synapses, right? So how you quantify synaptic specificity, so that's what we're trying to figure out too, right? Like, it's not just a graph, right? We need, I think, just a different way to define synaptic specificity, at least in the context of our problem.
I see. So just to summarize, so you're saying that there's some computational questions, which is how do you define some of these specificities, but there's also a biological data question, which is if the specification happens in spatially subdefined subregions of the brain, then, right, like it's the case that one cell type will never connect to another cell type, but it's not necessary for that to be genetically determined because in their spatial overlap, there's just no chance those would connect. And then there's a spatial information basically there as well. I see. Okay, but it seems like it could be tried, is what
of course.
it would.
I mean, if
and
you take
it's
a
not.
connectome, somehow subtract the spatial information, and figure out how to define the choice.
Yeah.
I think this
one should be able to do this.
So
that seems amazing. Yeah.
In the back?
Yes.
Hi, thank you for your talk. I might have missed this from earlier in the talk. I was wondering, do you have, like, given all the information you have now, some way to understand, like, maybe a temporal sequence of decisions that the cells make?
We can't.
I mean, are we sure? Because, like, that would imply that we definitely need to do, like, time point kind of experiments. Because I was just wondering if...
Yeah, I don't know. Maybe I don't know how to do it. So, like, again, coming to this sort of temporal sequences, spatial restriction, right? Why this case worked, essentially, we kind of removed all these variables, right? We picked the closely related neurons in the same space, and we put most of the variables sort of the same, right? Constant, most of the parameters. And that's why we kind of figured out some specific predictions. But like, you're saying, like, can we just, looking at the transcriptome, predict this temporal
Like,
order of wiring?
for example, if a decision is, like, broader, that could, you know, conceivably happen earlier in time. And the more specific the decision, maybe that happens later in time. I mean, just ideas like this.
No,
That-
we know that it has to be like that for the connectome side, right? You need to go from broader choice. You're born and you can go anywhere. You project a particular part of the brain. Now your choice is not 8,000 cell types, it's 1,000 cell types. Then you form more spatial organization, right? Now you have choice only 100 neurons, then 10 neurons, you know, that's how it happens probably. I mean, it has to happen like that. Yeah.
I think we're out of time, so thank you so much.
Thank you.